Guys, welcome back to Myaba Academy. Okay, so we're still on the roll and we're trying to figure out how to understand or assume the pathogenicity of variants using multiple in silico tools. So in my previous two videos we checked the pathogenicity using variant effect predictor, and then we used polyfen 2. So those were for exonic regions. Those were for genes that can produce proteins. So what do we do when we have a mutation in introns, those regions that don't code for proteins? So for that we have this very widely and highly used Splice AI tool. So this is the only tool for Splice AI that I would recommend using. So what it does is that when we have a pre-messenger RNA that is going to be translated into proteins, it has to be spliced. It has to get rid of introns. So to get rid of introns, splicing is performed. But when you have a mutation in your sequence, the splicing can go wrong, resulting in abnormal proteins, or maybe just proteins that don't work, that don't form. Okay, so for this we have example I picked from that website. Okay, moving over to the website, we come here. Okay, so if you want to look for the website, this is the name. Even if you write this is the first thing that comes up by Broad Institute. That's how easy it is to look it up. Okay, so not to be confused with other Splice AI, because there is another AI apparently that is for sound and other stuff. That's not what we're using today. Okay, so I read this paper when I was going through this website, and I think it's very helpful in understanding how this AI works. So when you open the paper, you come across a picture that is very interesting. So I came across this picture of this gene. Basically they have compared, so what this AI does is that it takes the normal gene and it takes the variant. So here, as we can see, the G is converted into A, so it goes into the Splice AI deep neural network. Splice prediction occurs, and as you can see here, this is the score of the normal protein. Sorry, so the normal score is 0.92, but as you can see, there is a gain in the variant. So this shows through deep neural networks that we can predict whether splicing will be normal or not. So as you can see here, that it is very confident in the fact that splicing will not be normal and there will be a gain. So yeah, this is how it basically works. As we go down, we come across this example format. This is the way you are supposed to write. These are multiple ways you can write your genomic coordinates, or maybe NMID with the name of the gene. So I prefer using this method because it seems the easiest. We'll keep it to the modern HG38 version, basic, and refinal scores. So loading. So yeah, it's that easy to get scores. Okay, so as you can see, here's the details of the gene. So apparently, as we can see, is that it is a stop gained mutation. This means that splicing does not occur. So here are the scores, and moving over to the first score, as we can see, the score is 0.0. This predicts the probability that the variant will cause the loss of the original acceptor splice site, meaning that there is no predicted loss. And as we can see, the score is 0.72 and it's yellow. That highlights the significance. that means that the variant will cause the loss of the original donor splice site at the 5' end of the intron. That means it strongly suggests that there will be a disruption in the existing donor splice site. And as you can see, the scores are 0 here, meaning that there is no predicted gain. And lastly, this predicts that there will be a new splice site. So, but the probability is so low that we would assume that this is our significant result. So this indicates that there will be a donor loss, and there will be a disruption of the splice site that leads to a frame shift mutation or the introduction of a premature stop codon, as we can see here. So we're sure that it would be a disruption in the formation of the protein. So yeah, this is how you use Splice AI for your variants. So yeah. Okay, there is one other thing that Splice AI doesn't do, is that it does not when we have insertions and deletions at the same time, it does not compute scores for it. So for that, it has another own custom version. This is the first version that you go here, and it's present all the way down. If you copy this code and enter it into Google Colab, you can insert your own sequences, and you would get your scores. So yeah, I hope it was a bit helpful, and yeah, see you in the next video, because I think we will learn about using other tools, and we'll also learn about conservation status and analysis. So yeah, thank you.